Welcome to Lecture 6A, the first talk for Week 6 of this course. This module will cover experimental tools and techniques used in immunology research, and we will start today by talking about the different types of experimental models that are typically used for immunological research, as well as the two main categories of cells used for cell culture or in vitro experiments, which are primary cells and immortalized cell lines. There are two other lectures for Week 6, one that will focus on the applications of antibodies in research, and one that will discuss one of the primary animal models used for immunology research, which is lab mice. As I'm sure you are all well aware, you have a midterm exam due at the end of Week 6. So there will be no extra credit quiz or discussion board this week in order to give you some extra time to focus on your test. As a quick note, these lectures for Week 6 and Week 7 will all relate to experimental and diagnostic tools, which all kind of fall together under the same umbrella of research. And so the lectures for these 2 weeks will fit together well and then also generally be a bit shorter and not as dense as the previous weeks of applied immunology. So just note that these next 2 weeks worth of lecture material should give you a little bit of breathing room after some pretty busy weeks with dense lectures as well as the midterm exam. In lectures this week, we will talk about tools and techniques related to two main categories of experiments. The first are named in vivo experiments, which is Latin for within the living. And as a side note, whenever you're using Latin terms for naming things in biology, such as the term in vivo or the names of bacterial species, which we've seen before, it's a common convention to italicize these terms. Now, since in vivo means within the living, it refers to studies that are performed using a whole animal, usually a model organism. I'm sure most of you are familiar with the concept of model organisms if you've taken biology courses before, but model organisms are species that have been widely used in biological research, including things like mice, zebrafish, as well as fruit flies or Drosophila. These species are used with the expectation that certain conclusions drawn from experiments in model organisms can then be applied towards our understanding of biological processes in other species that can't be used in experiments, namely humans. Model organisms are usually inbred, meaning that it's easy to breed or purchase many individuals who are genetically identical to one another. They are also relatively easy to maintain as an animal colony in a lab setting and can be genetically manipulated in order to study the effects of deficiency in certain pathways or cell types. We will talk a lot more about model organisms in our upcoming lecture on mouse models, since lab mice are one of the most important models used in immunology research. The other main type of experiment is the focus of this current lecture, and we call these in vitro experiments. In vitro is Latin for within the glass and refers to experiments where cells are isolated from tissues and then grown in a plastic dish, and this is what glass refers to. In vitro is also alternatively referred to as cell culture or tissue culture, so we can use these terms kind of interchangeably. Here, rather than studying an entire animal, individual cells are plated onto sterile cell culture surfaces, which are usually a tissue culture plate or a flask, and they are plated with cell culture media, which is usually this bright red color, and this is just liquid that contains things that the cells need to grow, like a food source, which is often glucose, as well as other nutrients and amino acids. There's usually some buffering reagents to help keep the pH stable, as well as other supplements. The specific media formulation and supplement composition depends on the cell type being cultured, since different cells have different requirements for their growth and survival. These three things are combined together and then cultured under sterile growth conditions. And this sterility is very important, since contamination of cell cultures with bacteria or fungi is a very common mistake in cell culture if people do not use correct sterile techniques in a tissue culture hood. A pretty standard incubation for mammalian cells is at 37 degrees Celsius, which is body temperature, with 5% carbon dioxide, although again, like media, these settings are really dependent on the type of cells being cultured. These conditions are maintained in an incubator, which is an example shown here on the right, which has shelves for holding plates, and it keeps things like temperature, carbon dioxide, and humidity levels constant at whatever settings the cells need. In addition to in vivo and in vitro experiments, there are a couple other categories of experiments. In situ translates to within its original place, which refers to biological observations that are made using cells that are within the same location that they were in a living animal. So in situ applies to certain types of imaging or even spatial transcriptional analyses that were performed on tissue sections where tissue or organ architecture has been preserved. And this gives you the advantage of providing some spatial information about certain biological measurements. We will talk about in situ analysis a little bit more when we discuss the use of antibodies when applied for imaging tissue sections. You may also see the term in silico being used, which is actually pseudo-Latin and just refers to in silicon, as in the silicon that's present in computer chips. I think that this is a little bit lame because you could have just used other existing terms for this instead of fake Latin, but whatever. So in silico refers to computational biology, which is also called bioinformatics. And this is where scientists either create models or analyze massive data sets from in vivo, in vitro, or in situ experiments, where traditional forms of analysis would be insufficient. And this applies to things like transcriptional profiling, which is also called transcriptomics, or genome profiling, which is also called genomics. Again, we won't discuss these other categories at length in applied immunology. Now, given these different types of experiments that could be conducted in order to examine certain research questions, what are the relative advantages and disadvantages, specifically comparing in vivo versus in vitro models? I think that comparing each type of experimental system across metrics like cost, ease of manipulation, and physiological relevance is a really helpful way to place these systems in context before we drill down into the in vitro models that we will discuss today. Let's compare in vivo versus in vitro models by these metrics listed in the left-hand column, just by going through each one by one. To start off, the costs associated with obtaining and maintaining each model are very different, and this is a very important consideration, especially for smaller labs or those at institutions that don't get as much funding for their research. So animal models like mice are considerably more expensive to obtain and then care for on a daily basis through something that we refer to as animal husbandry. They need to be cared for, bred, genotyped, and then used for experiments where the types of analysis tend to be a bit more expensive. Mouse experiments tend to take longer as well, since the animals need to be housed up to a certain age for experiments, and this might take longer if you're studying things like immune memory. Cells, on the other hand, are relatively cheap, since all you need are cell culture reagents and incubator space. At least this is cheaper for basic types of cells, though obviously there are some special types of cells that require expensive media formulations. The types of analysis that are typically performed on in vitro cell cultures also tends to be less expensive compared to mice, though again, this is a generalization, and things like transcriptional sequencing of cultured cells are obviously quite expensive. Next, let's look at regulation and ethics of each model. Use of animals in research is very highly regulated for obvious reasons, since it is extremely important to ensure that principles of animal welfare and ethics are being followed when working with animals, and this is done in order to minimize harm to living creatures. Related to these principles, researchers also try to minimize the number of animals that are used in experiments by designing experiments that should work based on previous observations, as well as predicting the number of animals needed in order to generate convincing statistics given a certain effect size. Obtaining human cells is also highly regulated, as there are many important ethical concerns related to patients that need to be taken into consideration. There are also some additional protocols that must be followed when handling human cell samples compared to those from other species. But in general, once cells are obtained, they are much less laborious to work with on a regulatory front, since their daily maintenance and culture is not subjected to the same level of scrutiny as it is with live animals. These models also differ with respect to levels of technical skill needed to use them. So mouse models usually require anywhere from 1 to 2 years of training lab personnel in order to really gain true proficiency with handling and manipulating mice, as well as in the protocols related to the processing of tissues that are obtained from terminal harvests. Technical proficiency with animal models is extremely important in order to minimize the introduction of technical variability between different animals, since mouse experiments usually have more variability in their data sets to begin with, just given the complications of working with live animals that are influenced by environmental factors. In contrast, many in vitro techniques are generally much easier for new students and scientists to learn. This statement really applies to basic techniques though, so obviously there are some much more sophisticated forms of cell culture and analysis that take a while to learn, and I don't mean to minimize that. But in general, once a trainee learns sterile techniques within a few months, they can culture lots of commonly used cell lines. It's also important because in vitro cell culture is less expensive compared to animal work, and so the mistakes that someone inevitably makes during their training period end up costing less money when you're working with cells compared to mice. Beyond those first few metrics, we can also compare these models across things that actually affect the types of experiments that you can do with each one, starting with the ability to genetically manipulate mice and cells. Part of why mice are such a popular model organism is because there are extensive genetic tools available in terms of mouse strains that lack certain genes, overexpress other genes, or have things like fluorescent reporters that are related to a given pathway. But if you have to either make new genetic tools for mice or breed mice in order to create certain combinations of these targets, this can take a really long time and therefore end up costing a lot of money. Also note that getting cell-specific or tissue-specific genetic targeting is a lot more difficult in mice, and although there are some systems that work quite well, which we will learn about more during lecture 6C, a lot of these are not 100% specific for a cell or tissue of interest. Cultured cells offer a real advantage in that they are much easier to genetically manipulate. There are well-established protocols for the artificial expression of target genes or modified genes through things like retroviral or lentiviral transduction, as well as tools for knocking out targets through CRISPR-Cas9 technology, in addition to genetic reporter systems for certain pathways. These protocols are constantly getting optimized by companies and research labs in order to create next-generation tools for expanding and improving the types of experiments that are possible with cultured cells. The ease of being able to genetically alter cultured cells in a relatively fast and cheap way is a real advantage of using in vitro systems. Another category of experimental treatment or manipulation are those that are pharmacological, or those that would involve administering drugs to mice or cells. In mice, drug treatments usually need to be administered in high doses in order to get good bioavailability, which means the effective concentration of a given drug that would be needed to actually act on target cells. Sometimes the pharmacokinetics of certain drugs hasn't been well characterized for in vivo use, so drug dosing regimens for a given experiment might need to go through several iterations in order for them to be optimized. Drug treatments may also have off-target effects in animals, since these are a much more complicated biological system compared to the relatively simple isolation of cells in a dish. So if a drug has off-target effects on other cell types in the body, it might confound the interpretation of data from a given mouse experiment. Cultured cells are generally easier to manipulate with pharmacological treatments. Since there aren't issues with route of administration or organismal metabolism, drugs are typically absorbed by cultured cells more easily, and it is much more straightforward to calculate effective concentrations that are acting on cells. Because cells are usually cultured as only one or two cell types together in a dish, this really helps to prevent any confounding observations that result from off-target effects. So it's more convincing that an observed effect following a drug treatment is truly due to biological processes that are occurring within that one cell type. Physiological relevance is a really important metric to consider as well. So far, animal models have established a lot of disadvantages related to their cost and ease of manipulation through experimental treatments. But despite these disadvantages, animal models are really a representation of the best type of model for answering scientific questions that may involve multiple cell types whose functions must be coordinated both within and across different tissues. This is really important for things like immunology research, where so far in this course, we've learned that immune responses are really coordinated on the level of an entire organism rather than on a small isolated population of cells. Obviously, in vitro cell culture models lack this level of information, since studying cells in isolation means that you are missing contributions of other tissue or organism level effects on the types of responses that you measure in a given cell type. So in vitro models can run the risk of having cells that may not function in the exact same way that they do physiologically within an entire organism. Because in vitro systems are so easy to manipulate in various ways, there's also the risk of being able to sort of push systems beyond what is physiologically relevant, and therefore showing experimental readouts or functions of cells in vitro that might not actually be observed ever in a physiological setting. With that in mind, in vitro systems are still extremely valuable and advantageous in immunology and biomedical research as a whole. So because cell culture is such a reductionist system, in vitro experiments allow us to perform highly controlled studies on individual molecules, pathways, and cells in ways that just aren't possible in whole tissues or animals. So in vitro models are crucial since they can be used for rigorous experimental testing that allows for the determination of signaling and cellular pathways that underlie a biological observation, which in research is often defined as the underlying mechanism. As for in vivo models, it should be obvious at this point that their primary advantage is that they can test the contribution of individual molecules, pathways, or cells of interest towards an animal's actual response to a given disease or treatment. So, animal models are valuable in providing evidence of organism-level responses, which are also referred to as phenotypes. Given that immunology research is usually done with the goal of translating preclinical findings into new therapies for human diseases, conducting research in animal models is an extremely important step in this process before pursuing clinical applications of a research finding for human medicine. With these comparisons, I want you to understand that neither one of these models is inherently better than the other. So any experimental model comes with its own set of drawbacks or caveats. And the important thing is to acknowledge the limitations of each of these tools and not overstate the conclusions that are drawn from individual types of experiments. Overall, good experimental design requires balancing the overall physiological relevance of a given model with its ability to let you convincingly test a given scientific question. Usually in immunology research, a comprehensive research project will involve both in vitro and in vivo experiments, which provide important information on both the mechanism and the physiological relevance of a given pathway or cell type that's being studied. So just keep this information in mind, both for the remainder of this lecture on in vitro cells, as well as during lecture 6C, where we will learn more about mouse models that are used in research. For the remainder of this lecture, we will learn more about the types of cells that you can work with in vitro. These are broadly split into two categories, primary cells and immortalized cells. We will learn more details about each of these in the next slides, but let's first go over a quick summary of what defines each of these categories. We can look at primary cells first, which are simply cells that have been isolated directly from tissues following tissue dissociation or cellular purification steps. These can be obtained in a number of ways from both humans and model organisms like mice, though the availability of certain tissues from humans is obviously much more restricted. Today's summary will obviously center on different types of immune cells since, not surprisingly, we've discussed these the most in an immunology class, but keep in mind that these comparisons hold true for many other cell types including fibroblasts, epithelial cells, and tumor cells. So first, primary immune cells can most easily be extracted from tissues where we expect to find high concentrations of leukocytes. And this includes secondary lymphoid organs like the spleen and lymph node, as well as leukocytes that are found circulating in blood. Primary human immune cells are easiest to isolate from blood since a blood sample is relatively easy to collect and is relatively much less invasive compared to getting tissue biopsies from whole organs. For blood and secondary lymphoid organs that are collected from mice, minimal tissue processing through mechanical dissociation and cell isolation can yield high numbers of immune cells for experiments, including T and B lymphocytes, as well as monocytes and neutrophils. Primary immune cells can also be derived from hematopoietic stem cells that are found in the bone marrow. This is obviously something that is much more commonly used in model organisms compared to humans. So we know that the bone marrow contains HSCs and that these can give rise to all lineages of immune cells. So if you have bone marrow from a mouse, you can plate those cells in culture with various cytokines, and the HSCs in the bone marrow will differentiate into your immune cell type of choice. One really common protocol used by innate immunologists is to differentiate APC subsets such as macrophages and dendritic cells, which are referred to as bone marrow-derived macrophages, or BMDMs, or bone marrow-derived dendritic cells, or BMDCs. These cells aren't isolated in their mature form directly from the tissue itself, but since they are differentiated from primary stem cells, they are still considered primary cells. Lastly, mature immune cells can be isolated from non-lymphoid tissues such as the lung. These protocols usually require pretty aggressive tissue digestion protocols with a variety of enzymes and mechanical dissociation, as well as purification steps to further isolate leukocytes such as alveolar macrophages from non-immune stromal cells. This is different than processing things like the spleen or blood, where you can expect to get high yields of immune cells, so non-lymphoid tissues instead contain lower numbers of mature tissue-resident leukocytes, and these in general tend to not survive as well once they're taken out of their tissue of residence. In general, primary cells retain more characteristics of their cellular identity, although they do have some drawbacks that we will discuss shortly. The second category of in vitro cells are immortalized cells. These are actually derived from primary cells originally, which were then treated with one of several possible immortalization protocols. These include infections with oncogenic viruses that encode oncoproteins, such as SV40, which encodes something called large T antigen. Human papillomaviruses, or HPV, encode the products E6 and E7, which are also used for immortalization. Each of these are oncoproteins, which means that during the course of natural viral infection, they can cause cancer in infected tissues. SV40 large T does this by inactivating the tumor suppressor protein P53, while HPV E6 increases P53 turnover, and E7 inhibits another tumor suppressor called retinoblastoma, or RB. Interestingly, this activity by HPV is the underlying reason why HPV infection can lead to the development of cervical cancer later in life. Therefore, infection of primary cells with these oncogenic viruses can be used to generate an immortalized version of that primary cell, which is essentially now just a cancerous version of that primary cell. Another technique used to immortalize cells is by expressing a protein called telomerase reverse transcriptase, which functions by preventing telomere shortening. So the ends of telomeres usually become more truncated over time and eventually induce a state called replicative senescence in aged cells that makes it so that the cell doesn't divide anymore and often will exhibit altered cellular functions. By preserving telomere length, TERT therefore functions by eliminating a key regulatory process in cellular aging. The goal of all of these protocols is to generate a new form of the original cell that has now acquired the ability to proliferate indefinitely, and this is in comparison to primary cells, which can undergo only a finite amount of cell divisions before undergoing senescence. This makes immortalized cells much easier and cheaper to maintain and work with, and there are numerous commercial and academic repositories of cell lines that have been generated by different groups. However, immortalized cell lines come with their own drawbacks as well, so let's learn a little bit more about the advantages and disadvantages of working with either type of in vitro cell culture. First and foremost, primary cells offer the most physiological or faithful representation of cellular characteristics and functions compared to what you might expect to see in vivo. And this is because for many cells, they maintain more of their original cellular characteristics even when they're cultured ex vivo, at least in comparison to cell lines. Additionally, if you are working with primary cells from human donors, you are given additional layers of information about their biological characteristics. And this is properties such as age, sex, and medical history or disease status. And this can help researchers stratify resulting data sets to see if there are any sort of variable readouts that might be correlated with properties such as donor sex. However, primary cells do come with a number of disadvantages. First, primary cells exhibit finite survival when taken out of tissues, which we also refer to as a state of being ex vivo. This is because primary cells either survive poorly once they're taken out of an organ, which is the case with mature tissue-resident cells such as alveolar macrophages, or because they only undergo a finite number of cell divisions before becoming senescent. They are also less amenable to stable genetic manipulation, so certain types of protocols related to genetic overexpression or knockout tend to be less efficient in primary cells compared to immortalized ones. For rarer cell populations, such as dendritic cells that are isolated from tumor tissue, you also get very low cell yields, so you might need a lot of tissue in order to get enough cells to do an experiment. Lots of primary cells also don't have good viability if you try to freeze them for long-term storage in liquid nitrogen and then thaw them out for a future experiment. So you usually need to start using them the same day that you harvest tissues, which for some isolation protocols can take a lot of time. Lastly, human primary cells can be quite difficult from an accessibility standpoint, so you either need to get tissue from someone who runs a clinical research lab that has access to patient samples, or you can actually buy some types of primary cells through commercial vendors, but these tend to be quite expensive and not have quite as many choices. For clinical research, there are a lot of regulations related to the collection and access of primary human tissue, all of which relate to important ethical principles related to patient consent and de-identification. Also, for rarer samples like organ biopsies or tumor resections, researchers must come in whenever samples are available, and this is determined by patient surgery schedules, and this can make research scheduling a little bit more difficult. In conclusion, primary cells have important advantages related to how well they recapitulate what you might expect to see with cells under physiological contexts. However, they still come with some limitations that make them a bit more difficult to work with on a regular basis. Immortalized cells address a lot of the difficulties associated with working on primary cells. Because these are immortalized cell lines that don't require handling of patient tissues, they are not subjected to the same degree of regulation compared to primary cells. This is helpful for smaller labs or those that aren't affiliated with a clinical research department or large hospital, and so they don't have access to patient samples. There are many well-characterized cell lines used in immunology and biomedical research in general, and most of these are accepted in many types of scientific publications. Immortalized cells can usually be obtained from commercial vendors as a one-time purchase, and this is because they can then be expanded in culture and then frozen back for storage for future experiments, where, unlike primary cells, they can usually be thawed out with good viability. Immortalized cells are also generally more amenable to stable genetic manipulation through protocols like retroviral transduction or CRISPR-Cas9 mediated genetic knockout. Since these cells can be frozen back as stalks, you can therefore generate a series of stably modified cell lines at one time and then use those for the duration of an entire project. So in general, immortalized cells tend to be a lot easier to work with on a regular basis compared to many different types of primary cells. However, these advantages usually come at the price of being a potentially less physiologically relevant in vitro model. And this is because the process of immortalization essentially generates a cancerous version of the initial primary cell. And although this allows for unlimited replication, it can still obviously influence other cellular properties. So, immortalized cells often exhibit a loss of some cellular characteristics, including those that are associated with cell identity. Additionally, the process of repeated cell division and culture, which we call serial passaging, can lead to the accumulation of both genotypic and phenotypic alterations to that cell line over time. So this might not be a problem over the course of a few weeks, but consider that some academic labs have used the same cell line source for decades, in which case it's easier to see how that line could essentially undergo genetic drift compared to its initial population. Collectively, these issues mean that immortalized cells run the risk of providing a less faithful representation of that cell's actual physiological functions in vivo. So, although immortalized cells are in general much easier to work with, they may introduce additional confounding factors or caveats that are worth considering when drawing conclusions from data that's obtained using them for in vitro experiments. And it's very important when you're deciding which type of in vitro cells to work with for a project to factor these sorts of concerns into your experimental design process. Today we've learned about how in vitro cell culture models allow scientists, such as immunologists, to test experimental questions in a reductionist or mechanistic level, and that the information that we get from in vitro experiments can really nicely complement data from other more physiological models, such as those performed in vivo. We established that in vitro and in vivo experimental models each come with their own pros and cons, and that immunology research often requires data gained from both in vitro and in vivo experiments in order to convincingly support a researcher's scientific conclusions. We subdivided in vitro models into two different categories of cells, primary cells and immortalized cell lines. These differ from one another with respect to how they are derived and many factors related to the overall ease and use of each model. In general, primary cells can be more expensive and difficult to work with compared to immortalized cell lines, but they often provide a more physiologically relevant cell type that maintains aspects of its cellular identity compared to cells that have been subjected to immortalization protocols. That's it for today's lecture, but remember to watch the 2 remaining lectures for week 6, which will cover uses of antibodies in research protocols, as well as lab mice, which are probably the most commonly used in vivo model for immunology research.